Welcome, biology students. This is a quick video about lesson 2.2 or assignment 2.2, the Mitosis online lab. First thing you're going to do is go to the lessons. You can check out the assignment in assignment section, but we're going to go through the lessons to give us some background in all of it. And we're going to scroll through all the way to this 2.2 Mitosis online lab. As we click on it, it's going to take us to the actual lab. Please note that it's 60 points. The first part is going to be 40 points and the analysis is going to be 20 points. So we click on the online lab, and the first thing that you'll notice is there's a picture here of different cells. These are all plant cells, and they're actually onion root tip cells because onion roots or any plant root is growing a lot, which means that there's a lot of cell division that's happening there to grow, for the plant to grow. So the cells, the root cells are dividing and dividing and dividing, which is what mitosis is. So that's why you might happen to see a lot more cells going through mitosis in root cells. And remember, these are plant cells, so they're kind of square looking because they have that cell wall. So the first slide we look at, we can see, if you remember from your modeling, the meat in the middle, and then also the metaphase, there's telophase here where they're kind of into two different nuclear areas. And then there's anaphase where the sister chromatids are being pulled apart. Okay, so there's another example here, but most of the cells are actually in interphase, performing their normal function. And then there's a few areas where there is prophase, where the chromosomes start to condense and start to continue with the example. So here's interphase. Normal functioning cell, you can see that most of the cells look like this. Then you have prophase, where the chromosomes start to condense. Then we have metaphase, where they meet in the middle. The centromeres are starting to attach to these sister chromatids and get ready to pull them apart, which happens in anaphase, where they start to be pulled apart. And then telophase is again where you see these different examples of the cells starting to reform and divide in two nuclear areas. All right, so those are the different examples. If we looked at slide 2, we have an example of the onion root tip again. You can see our anaphase where they're pulled apart, the meat in the middle, another anaphase. A lot of them are in interphase, as you can see, a lot of them. And a few are in prophase, but most are in interphase. And then again here we've got meat in the middle, metaphase, anaphase, and most of them again are in interphase, performing the normal functions of the cell. There are some where the chromosomes are starting to condense, and that's prophase. And finally, the last cell, as you can see, most of them are in interphase right here. And then you can start to see the chromosomes here, that's prophase, but most of them are in interphase. And then there's a few that are in metaphase, another here that's anaphase where it's being pulled apart. All right, so those are the actual observations that you're doing. And so for the activity, you're going to answer these questions. For each slide section, it's worth 10 points. So you answer the identifying characteristics, you draw the drawings, and you put that there. Then you count the number of cells for each of the slides and total. Make a drawing of the cell you see the most. Again, count the number of cells here, count the number of cells here in each of the different sections. And finally, for the analysis, using what you observed, you answer each of these 5 questions. Each of them are worth 4 points each. And that's it. So good luck.